All right, anatomy students, we're going to be looking at a review over the cell in this lecture, and we're going to talk about the organelles, the nucleus, nucleolus, cell membrane, mitochondria, Golgi, lysosomes, the ER, cytoplasm, cytoskeleton, vacuole, chloroplast, centrioles, and ribosomes. And this should be just a quick review because I know that we went in depth, especially in advanced biology class last year. So let's take a look a little bit more specifically at the various organelles. The nucleus, of course, you know, is the control center or the brain of the cell, and it controls all the cellular activities, and it houses the DNA, and the DNA is the deoxyribonucleic acid, and that's what makes up the genes that make you up and code for your characteristics and your traits. The nucleus has chromosomes in it. The chromosomes carry information to determine what traits a living thing will have. So here's the cell. Inside the nucleus are the chromosomes. These chromosomes are already wound into chromosomes that are visual. But if we could unwind these, we see a single strand of DNA. And sections of DNA that code for particular proteins are called genes. DNA stands for deoxyribonucleic acid. And like I said before, we usually see this in this double helix or the twisted ladder form. And we went into depth quite a bit last year on DNA and the structure of DNA and coding for proteins. We won't go that in depth with the anatomy. We will talk about some genetic disorders as we go throughout the systems, and those genetic disorders, of course, occur on this level, the molecular level of DNA. As I said before, DNA carries that genetic information, and there are thousands and thousands of genes, and it specifies everything that's needed in those genes to maintain and to function and replicate cells. It's made up of 4 different bases, and you're familiar with these, I'm sure. Adenine, cytosine, thymine, and guanine. And we always talked about how adenine and thymine pair together and cytosine and guanine pair together, the pyrimidines and the purines. And that's what we see here. Adenine and guanine are examples of purines, and thymine and cytosine are the pyrimidines. So we pair A and T, C and G. The nucleolus is found inside of the nucleus. It's usually seen as a dark spot if you look at it through a microscope, and it's tiny, and it's considered a nucleolus because it stands for a small nucleus inside of the nucleus, but it doesn't have all the responsibilities that a nucleus has. If you remember from last year, we talked about how the nucleus is where we see RNA being formed. So that's its main function, which is RNA is very necessary in making proteins and decoding the DNA. The cell membrane holds the cell together. And we talked last year about how it was a lipid bilayer. So that has 2 layers of molecules, and the layers are made up of phospholipids. And you can think of this as a sandwich with 2 pieces of bread and some of the stuffing on the inside. Kind of an odd analogy, but it works. A phospholipid, each phospholipid has 2 different ends to it. One end is hydrophobic and one end is hydrophilic. Hydrophobic means water-fearing, and hydrophilic means water-loving. So what happens, just like the sandwich, they turn tails. So the hydrophobic tails form this barrier, this waterproof barrier, and the hydrophilic heads, this is the outside of the cell and this is the inside of the cell or the cytoplasm, are attracted to the liquids on the inside and the outside of the cell, but that leaves this waterproof barrier in the middle. One end of the molecule is afraid, the other one loves the water, and proteins float throughout that, and it allows it to be a model called the fluid mosaic model, where these can float and move throughout this phospholipid bilayer. The mighty mitochondria. Mighty mitochondria are the powerhouses of the cell, and their whole job is to break down the energy that we eat when we take in foods, break those molecules down so that the cell has energy to live. Has 3 main parts, and it has an outer membrane and it has an inner membrane, and this is called the matrix, which is the liquid inside of it, and these folds are called the cristae. Glycolysis occurs in the inner membrane. The matrix is a fluid that has water and proteins all mixed together like a solution, and that's within the cristae of the mitochondria. Proteins take the food molecules in, combine them with oxygen, and then release energy. The endoplasmic reticulum is also shortened to be known as the ER. It's an organelle inside the cell that's made up of membranes that are in the cytoplasm of the cell. And there are two different kinds. We have rough and smooth. This guy over here to the right is pretty smooth, I guess. Rough ER has ribosomes on it, and the smooth does not. So the smooth ER, its main function is to collect, maintain, and transport things, and it's kind of tubular. And one of its jobs is also to create steroids. It stores ions for the cell to keep nutrients balanced as well. And like I said a minute ago, the rough ER has bumps all over it. That rough appearance is due to the presence of ribosomes. And the purpose of the ribosome is the site of protein making or protein synthesis. It says the ER collects the proteins built by the ribosomes and creates a bubble around them. This vesicle is formed when the ER pinches off a part of its membrane. Vesicles can then move to the Golgi body or the Golgi apparatus or the cell membrane. If the vesicle floats to the cell membrane, the proteins are going to be sent out of the cell through the process of exocytosis. And if they move to the Golgi, then the proteins are going to be used inside of the cell. So there's the smooth ER versus the rough ER, the ribosomes. This is three-dimensional, and you notice how this is actually, the rough ER is actually connected to the nuclear membrane, and we have it out here where we have no ribosomes and ribosomes here, so the rough and the smooth are actually attached to each other. Exocytosis is when we release things out of the cell, such as molecules like hormones or proteins that are too large just to go through the cell membrane. Osmosis, by definition, is the movement of water across the cell membrane from the area of higher concentration, which is here, to an area of lower concentration, which is here. We also refer to it as a special case of diffusion. The Golgi apparatus is also called the Golgi complex, and it kind of looks like a flattened stack of pancakes. And its job is to take in simple molecules, combine them to make larger ones, and take those larger molecules and put them into packages and secretes them, and it's called vesicles when they are secreted, packed. So this slide tells us to think about this example as the building of a model of a ship, and that's the molecule, and then when we take that model and we put it into a bottle, that's the vesicle. So it's just packaging the proteins or things, whatever has been made for transport. This diagram is embedded and shows you how that happens, just to give you a visual of those being encased in vesicles from the Golgi apparatus. Another organelle is called a lysosome, and these are primarily in animal cells, and we're going to find they're very important when it comes to human anatomy, especially when we look at infection-fighting cells. They combine with the food taken in by the cell, and the enzyme in the lysosomes bond to the food and digest it, so it has an acidic interior. And then smaller molecules are released, which are absorbed by the mitochondria. When an organelle no longer works, then the lysosome is going to attach itself to it. It's going to break it down like it would be food, so it's kind of like a recycling plant. They refer to it here as kind of like a cannibal, but it breaks it down so that it can be used in smaller parts someplace else. The chemicals then can be absorbed or excreted. Lysosomes are also known as a suicide sac, and ultraviolet light damages lysosome membranes. We have to be careful around those ultraviolet rays. The enzymes inside the lysosome would then spread throughout the cell and digest it, so it destroys the cell if it's accidentally opened up, especially by ultraviolet radiation. Here's what the structure of it looks like and how it works. All right, the cytoplasm, another word is called the protoplasm. It's everything inside the cell membrane. The cytoplasm itself is everything inside the cell membrane and outside of the nucleus except the cell's nucleus. So the protoplasm is everything, the cytoplasm is everything but the nucleus. The cytosol is made mostly of water, has organelles, and has salts and dissolved gases and nutrients in it. So this is a diagram indicating where the cytoplasm is. The cytoplasm is made up of the cytoskeleton, and we weren't aware that this was an organelle until a few years ago. So the whole function of the cytoskeleton is not only allows things to move throughout the cell, but it helps the cell to maintain its shape, and it keeps it from getting smashed or dented in when it's up against something else. So it allows it to retain some shape. So the cytoskeleton is made up of microtubules, let's go back to that, and microfilaments, and we're going to talk about that. You see it here in the structure. We're going to talk about the use of microtubules and microfilaments when we talk about muscle structure as well. In human anatomy, we don't talk much about the vacuole, Vacuoles are large in plant cells, but almost non-existent in animal cells, but they are bubbles, basically, that float inside the cell, and they're more important, like I said, to the survival of plant cells instead of animal cells, so we won't spend time on those. Centrioles are rather important to animal cells. We don't find them in plant cells, and they look like two cylinders at right angles to each other. We looked at them last year and said they look kind of like churros. So when they are viewed with an electron microscope, they show up as 9 bundles of microtubules, and they're arranged in a circle. And they help us to form fibers that move the chromosomes around when the cell goes through cell division. As animals prepare for cell division, these two centrioles separate, go to opposite ends of the cell. Then we have kinetochore fibers that are involved that will attach to the chromosomes at the centromere and take them back to the poles during anaphase. We're going to finish up the organelle review with ribosomes. These are small dot-like structures in the cells, and we talked about that when we looked at the endoplasmic reticulum. And they're often associated with forming the rough ER. They are the site of protein synthesis inside the cell, and they are made in the nucleus of the cell, actually in the nucleolus. And a ribosome can make the average protein in about 1 minute. Ribosome structure. Ribosomes are made up of proteins and RNA, or ribonucleic acid, and these molecules are arranged into 2 separate subunits. These subunits get attached to each other, and they form the entire ribosome, and when viewed through a light microscope, they appear as dots. They're actually 2 subunits together. There are 2 kinds of ribosomes. We have the ones that are attached to the rough ER, and some that are floating in the cytoplasm of the cell. The ones that are attached to the ER make proteins that are used in the ER or get transported within the ER. And the free ribosomes that make up proteins are used in the cytoplasm. So that's a really quick review of the organelles that we spent quite a while last year going over, and we're going to stop there today and continue with diffusion and osmosis in the next lecture.